everyone. Welcome again. We're making progress in ALS research at Everything ALS. It's what we do. We aim to discover new insights, improve early detection, and find a cure. And with that, it's over to you, Sarita.
Good evening, everybody. It's my pleasure to introduce Dr. Zimri and Dr. Tracik, our presenters for today. And a brief introduction to Dr. Zimri. She is a dedicated researcher, neurobiologist, and expert in preclinical drug development with over 12 years of experience in neurodegenerative disease research. As the Vice President of Research and Development at NeuroSense Therapeutics, a clinical stage company pioneering treatments for neurodegenerative diseases. She leads all preclinical projects, shaping scientific strategy and steering innovative therapies from concept through clinical application. She is a PhD graduate in neurobiology from Tel Aviv University. She's an active contributor to scientific and public health communities with roles on the bio community, We at Health Tech, Bashar, an academic society in Israel, and Medad, a nonprofit focused on promoting public health. Her dedication is driven by a commitment advancing solutions for critical challenges in neurodegenerative disease research, striving to bridge the gap between scientific discovery and patient care. It is also my immense pleasure to introduce Dr. Tracik. He is the Chief Medical Officer with over 20 years of experience in general management, medical affairs, clinical development, and commercialization in biotech industry. His experience and expertise in therapy is extensive and includes different disease areas such as CNS, respiratory, oncology, ophthalmology, infectious diseases, and transplantation, with a specific focus on neurodegenerative and autoimmune diseases. Before joining the pharmaceutical industry, Dr. Tracik worked at the University Clinics of Neurobiology at Clinic of Innsbruck. He holds a doctoral degree in human medicine from the Free University of Berlin. Thank you so much. Welcome.
So thank you so much for this very kind introduction. And we would firstly like to thank Indud and the amazing, really amazing team of Everything ALS for having us here. And it's always exciting for us to speak in front of the community. So whenever you call us, we come and we are in, and we are very excited to share with you the key findings and results from our phase 2b study Paradigm. Yeah, so friends, can you switch the slide? But first, since we are a public company, so these are our forward-looking statements, and we can begin. Yeah, so I mean, as we all know, the pathophysiology of ALS is very complex, and it's underlying multiple pathways and mechanisms that are involved. And therefore, we truly believe that in these complicated diseases, we need to change and shift the paradigm and not to think about a silver bullet rather than a combination therapy that can help us to create a maximal effect. So PrimeC, our lead candidate for ALS, is a tablet assembled of 2 FDA-approved drugs, celecoxib, which is a COX-2 inhibitor working on the neuroinflammatory processes, and ciprofloxacin, who is a fluoroquinolone affecting regulation of microRNA. And it's also an iron chelator, and therefore it's also affecting iron metabolism and iron pathway. So each one of the components targets several of these key pathologies, but importantly, the unique extended release formulation of PrimeC really allows us to maximize the synergism between these two components. Now, when we are thinking about combination therapy, I think that there are 2 advantages that can come to mind. The first one is that we want to combine 2 drugs together in order to improve the pharmacokinetics of the drug. Being in the drug development world, we know that one of the main challenges is actually this penetration to the brain and the effect on the brain. And in NeuroSense, we are able to show that using our preclinical studies and our PK profile studies. The second thing is that we want to improve the efficacy of the drug. And for that, we started with our path with a preclinical study that presented exactly that. So if we can move forward, thank you. So we conducted a preclinical study using zebrafish models. Zebrafish are very novel and unique models for drug screening. We had SOD1 mutant and also TDP-43, so the key main ones in two leading laboratories, one in McGill University in Canada and the second one in Israel. And as you can see here, when we examine the effect of the components by testing each one of the components and the combination, what you can see is that we see a significant improvement in the motor performance of these animals. So each one of the components gave us a beneficial effect, but we can clearly see the synergistic effect of these components. Actually, we also see that relative to riluzole, which is the standard of care. But the effect was not only obtained in the motor performance, but also in the morphology of the neuron. So in the right panel, you can see the reserved synaptic contact. So we can clearly see that following the treatment with PrimeC, we see a significant improvement in the connectivity of the neurons. And this effect was also obtained in supporting cells, such as the microglia. We published these results, these very exciting results in 2020. And that really allowed us to move forward into our next phase. But what we also did is to reinforce this result by using the same paradigm. So again, we used each one of the components and then the combination in order to demonstrate the synergistic effect for PrimeC. This time we did it in collaboration with Dr. Justin Hichida from USC, which is, I would say, the top leader in terms of research in iPSCs in ALS. And as you can see here on the left panel, we see the survival of the neurons. This blood was taken from ALS patient. It was then transformed into neurons, specifically to motor neurons, and then we treated them with each one of the components and then a combination and measured the survival of the neurons. So as you can see, we see a clear beneficial effect for each one of the components and a significant, in light blue, a significant effect for the combination actually rendered that to be similar to a healthy control, as you can see in the bars. Thank you. And what we also did is not only just to show the efficacy of the drug, but we really wanted to focus on the target engagement of the drug and really to show how this components actually affects the morphology and the pathology. And for that, we collaborated with Mass General Hospital and we conducted a 3-stage study using neuronal-derived exosomes. So neuronal-derived exosomes, just in short, is basically our vesicles that come from the CNS, and they go through the bloodstream into the periphery, into the blood, and basically allows us to measure parameters from the brain in real time in a non-invasive way. And we use this novel methodology to test a wide cassette of biomarkers related to PrimeC mode of action, such as neuron inflammation, iron accumulation, microRNA metabolism, and so forth. But also we tested several key hallmarks of the disease, such as TDP-43, which we all know is such an important aspect that we want to measure. And when we did so, we saw, first of all, that there is a basic phenomena for each one of these markers. But importantly, when we looked on the effect that was obtained in these markers using the blood from patients from our phase 2A study, which was an open label study with 12 months of dosing, as you can see on the right panel, we saw a significant decrease following the treatment in TDP-43 levels, but also in prostaglandin 2, which is the direct downstream mechanism for COX-2, which is the neuroinflammatory process. So these two significant reductions really suggested and reinforced the effect of PrimeC on these markers. And with this very promising result and the extensive preclinical study that have been done, we move forward for our phase 2 study paradigm. And Ferenc will tell you all about that.
Yeah, thank you, Shiran. So yeah, just let me say that I'm very pleased to be invited to your conference. And as Shiran said, whenever you call us, we are here, right? Even though it's it's not late, actually, it's early morning, right? But it's a pleasure, and I would say even an honor being with you guys and presenting our latest data. Yeah, as Shiran said, the paradigm study is actually built on all the knowledge and data we generated in our preclinical and clinical program. And the paradigm study is a phase 2b study. It's a randomized placebo-controlled double-blind multi-center trial. The study consists of 2 phases, a 6-month double-blind phase, which is followed up by a 12-month open-label extension phase. Today, I will present the 6-month data, meaning 6 months double-blind phase and the 12 months data. So 6 months double-blind phase and 6 months open-label extension. In 1 month, I hopefully can present the 18 months data, because we just are in the phase of the database lock, and we expect to have the top-line clinical results by mid of December. And by that, the study is also, let's say, officially concluded. And we enrolled ALS patients for this study, all forms of ALS were allowed, so sporadic and genetic forms, with a disease duration up to 30 months, and an upright slow vital capacity of 60% and above. Standard of care, riluzole and radicava or edaravone were allowed in this study. However, we were stratifying the patients according to the baseline therapy. So making sure that we have the same, let's say, amount of patients under riluzole or edaravone in the active and in the placebo arm. However, we had also some patients who were drug-free, so no riluzole or edaravone. However, we had only 7 patients who were drug naive when they entered the study. So 19% of the patients were co-treated with riluzole. We actually had only 1 patient treated with edaravone. The reason for that is that the study has been conducted in Israel, Europe, and Canada, and edaravone is not approved in Europe. The primary endpoint of this study is safety and tolerability, and the 2 biomarkers, hopefully proving the target engagement of our drug, TDP-43 and prostaglandin, Shiran was just talking about. However, we of course also looked into clinical outcome, such as ALSFRS, slow vital capacity, PROMIS-10, and other quality of life questionnaires, so overall survival and complication-free survival. The study was very well balanced, so actually we had a spot landing at baseline in regards of the ALSFRS total score. So the ALSFRS total was 37.5 in both treatment groups. But the study was also balanced to all the other baseline characteristics, such as age, gender, and so on. So as said, safety was one of the primary endpoints. And here you can see the safety, let's say, data after 6 months double-blind phase and the open-label extension of another 6 months. I won't go through all these data, but I think it's fair to say that if you look on the incidence of treatment emergent AEs and serious treatment emergent AEs, that PrimeC and placebo are actually more or less on that same level after 6 months and after 12 months. After 6 months, we had only 3 death cases, right? 2 under PrimeC, 1 under placebo. So this is the reason why we couldn't say much about survival. After 6 months, this changed dramatically. After 12 months, so we had here 7.5% death cases after 12 months and double after 6 months. So these are the clinical outcome after 6 months double-blind phase. As you can appreciate, after 4 months, we actually reached statistical significance. However, after 6 months, we saw a non-statistical significance with a p-value of 0.12. Nevertheless, the difference was 29.2% in the ALS-FRS total, which equals to an absolute difference of more than 2 points in the ALS-FRS. So, and now these are, let's say, the latest data we have. These are the 6 months data. And as you can appreciate, after 6 months, after 12 months, constant treatment, right, compared with the group who has been treated with placebo for the first 6 months and then switched to active treatment, we actually see a statistical significant difference of 36.6% with a p-value of 0.009. This actually is an absolute difference in the ALS-FRS total of more than 6 points, right? This pattern, right, is actually what I personally have seen many times in different clinical trials with drugs with a disease-modifying treatment effect, right? So actually, you could call this study design also a delayed start design, right? So this is actually for us a very encouraging result, because actually what you can see is that patients who have been on placebo for 6 months are not able to catch up with the patients who were constantly treated with PrimeC. So of course, very important to understand what is the effect of PrimeC on the 4 ALS-FRS subdomains, such as bulbar, respiratory, fine, and gross motor function. And as you can appreciate, we see a statistical significant difference in the bulbar function, which means speech, salivation, and swallowing, with a difference of almost 2 points and a p-value of 0.008. I think it's also notably important to emphasize that we see a positive effect across all 4 subdomains after 6 months and after 12 months of treatment. As said, we looked also in other clinical outcomes such as slow vital capacity, overall survival, the stage-free survival using King's and Meters, ALS complication-free survival, ventilation-free survival, and quality of life. And I think it's really exciting, or I would say, to see that we see a constant positive effect across all these secondary outcomes, right? And we didn't reach statistical significance. Nevertheless, I mean, the study was not powered to show statistical significance, neither in the ALS-FRS nor in the secondary outcomes. However, as said, we see a constant positive trend in all of these secondary outcomes. I want to highlight actually 2 outcomes, and this is overall survival. As said, after 6 months, we didn't have enough events, but after 12 months, we saw a 43% better survival under the constant treatment with PrimeC, and a 57% better ALS complication-free survival. This means patients needed less continuous ventilation, less hospitalization, and as said, less death cases due to ALS. We also see a positive trend in the PROMIS-10, which is a quality of life questionnaire. However, we also looked into biomarkers, and Shiran will introduce you some really exciting data using transferrin and ferritin as two, let's say, leading biomarkers of the iron metabolism.
Thank you, Ferenc. Yes, I think that one of the main advantages, at least for me, when we have phase 2 studies, is actually the ability for us to dive deep into the mode of action, and in the opportunity that we had with Paradigm was that we could include a wide cassette of biomarkers in very different and diversified assays. So we had iron tests, but we also had microRNA analysis and even a colorimetric measurements. So all together build us a really rich cassette of biomarkers that allows us to really understand better the mode of action of PrimeC. And as you can see here, we measure the levels of ferritin and transferrin, two key, as Ferenc said, two key biomarkers for iron metabolism. And what you can see really nicely on the upper panel, this is the longitudinal levels of transferrin and ferritin. So we see firstly that in our placebo-treated arm in gray, we see a pattern which is very similar to what we see in the population of people living with ALS, meaning we see lower levels of transferrin that actually keeps going down with the progression of the disease. And in turns, we see an elevation in the levels of ferritin, and it goes with the progression of the disease. And as you can see in blue, following the treatment of PrimeC in the group that was treated with PrimeC, from the beginning, we see a stability in transferrin and a reduction in ferritin. And what we also did is to try and see if this effect actually correlates also with the progression of the disease by looking on the correlation to the ALS-FRS-R functional rating scale. And as you can see in the lower panel, we see the correlation of transferrin. So we see, as I said, the reduction goes hand to hand with the progression, and this was abolished following the treatment. And in ferritin, it even reached a statistically significant effect. So we see the baseline effect. So we see the elevation following the treatment, the progression of the disease, and the abolishment following the treatment. And that was very much encouraging because it actually suggests that indeed we see biological activity here for PrimeC that is driven by the mechanism of action which we hypothesize about. And what we also did, and this is very brand new, so this is kind of like a work in progress, is that we, as I said, also measure levels of microRNA, and we're looking on the pattern and the changes in microRNA effect. And Ferenc, if you can move forward to the next slide. Thanks. So as I said, this is work in progress, and hopefully we can come again and show more data. But I have to say that we are very excited about these results because basically also here we see no changes in terms of effect on microRNA in the placebo arm, and a significant effect following the treatment on regulation of microRNA. As you can see, we see a lot of microRNAs that are changing following the treatment. And specifically, when we are looking on the specific microRNA that takes part in this process, we identify a lot of very interesting specific markers, such as miR-199A5P, for example, that is involved in the progression of the disease, and it also correlates to ALS-FRS-R rating scale, so as well as miR-151. So there's a lot of indicators that are very interesting and very closely correlates to the pathology of the disease and relates to effect on neurons and oxidative stress and neuroinflammation. And as said, this is kind of like work in progress, and we are very much looking forward to sharing more data soon about it.
So in summary, we can say that the PARADIGM study showed a favorable safety and tolerability profile, achieving the primary endpoint. As you might have noticed, we didn't present TDP-43 and prostaglandin 2 data. This is work in progress, right? Because we wanted to do this to evaluate both biomarkers by end of the study. And as I said, the study has last patient out was a few weeks ago. So this is work in progress. And as soon as we have this data, we are, of course, more than happy to share these data also with you. And we saw an improved disease progression after 6 months by 29.2% and a statistical significant change after 4 months. And after 12 months, a difference of 36.6% in the ALS-FRS total, which led to a statistical significant p-value of 0.009. PrimeC slowed functional decline across multiple ALS-FRS subdomains. As said, the most pronounced effect was on the bulbar function. And PrimeC demonstrated a potential better survival after 12 months with a hazard ratio of 0.57, which means a 43% better chance of survival after 12 months. As said, we saw a consistent positive trends across all clinical outcomes, primary and secondary. And as Shiran presented, PrimeC demonstrated a modulation, a positive modulation of transferrin and ferritin levels, alongside with a significant impact of the regulation of key microRNA associated with ALS progression. So all in all, as said, we think these results suggest the disease-modifying effect of PrimeC, positioning it as a potential first-line treatment for ALS. So based on these data, we are in the process, it's not final yet, we are in the process to design a phase 3 study. We had an amazing opportunity to discuss this phase 3 study with the FDA. We had a Type C meeting on November the 6th. And what I can reveal is that let's say, that the FDA agreed on the principle of our phase 3 study, which is great. So the phase 3 study we are planning is a randomized, multi-center, multinational, prospective, double-blind, placebo-controlled study with a 12-month double-blind phase, followed up by 12 months open-label extension. The primary endpoint gonna be a joint assessment of function and survival after 12 months of treatment. And safety and tolerability, So what we proposed to the FDA is a so-called map. So it's so the ALS-FRS adjusted by mortality. Okay, so a mortality adjusted performance as the primary endpoint. And the FDA liked it. Of course, now we are, let's say, finalizing the protocol. We have to submit some simulations, but in principle, the FDA agreed on using MAP as a primary endpoint. The population is gonna be more or less the same as in our PARADIGM study. So male and female adults between 18 and 75 years of age with familial or sporadic ALS according to the revised LS-Correal criteria, probable or definite. And we will change the inclusion criteria only in one point or in one criterion, and this is the disease duration. As you might remember, for the paradigm study, patients were eligible with a disease duration up to 30 months. For our phase 3 study, we will, let's say, reduce the disease duration to 18 months. The patients will be randomized in a 2-to-1 ratio in favor of PrimeC. We will stratify the patients according to TRICAL's risk profile. That's also what we have done in the paradigm study, and ALS background treatment, such as riluzole, edaravone versus no background treatment. The patients will be assessed every 2 months using the ALS-FRS, slow vital capacity, survival metrics, quality of life, and so on. And of course, we will also look into adverse events. We will do, as we have done in the paradigm study, a extensive lab control and ECG monitoring. So now, as said, we are more or less only 3 weeks ago, 3 weeks, we're gonna have the paradigm 18 months data, which is, as said, then also the end of this study. We will hopefully have soon also the additional paradigm biomarker results, not just as presented by Shiran, the microRNA, also neurofilament, and hopefully TDP-43 and prostaglandin 2. In 2025, we plan to have a pre-NDS meeting with Health Canada in order to discuss the filing for a conditional approval in Canada. We hope to submit for an NDS in Q2 2025. We're gonna meet the EMA for an ALS phase 3 study, or they call it a scientific advice meeting in Q2. And the plan is to enroll first patient in our phase 3 study in June, July 2025. By the way, we plan to conduct a study in US, Canada, Europe, and Israel. The current sample size estimation is 300 patients. But what we discussed on our meeting with the FDA, we're gonna use a so-called adaptive design. So we will have multiple check-ins during the study in order to control the variability of the study. So if needed, we can, let's say, enroll more patients, or if not needed, we can actually stop the enrollment even earlier than 300 patients. And we are also conducting a study in Alzheimer's disease, also with PrimeC. So with that, I want to thank you for your attention. But I mean, all these efforts and achievements wouldn't be possible without the strong support of patients. We are incredibly thankful that that patients are supporting us, not just, let's say, mentally, not just emotionally, also by participating in our study. Yeah, this is outstanding. And as Shiran said, she was the employee number 2. Now we have 17 employees globally, right? So we are still a very small company, but with the start of the phase 3 study, as said, of course, also in the US, we hopefully will grow substantially. And that's it from my end. Well, we are both more than happy to take any questions. Thank you.
Thank you. Thank you, Shiran and Frank, for this excellent presentation. And if you stop the presentation, we will go into the Q&A. And the first section for the first question was, will NeuroSense apply for EAP program, or will you offer expanded access program?
This is a very good, excellent question. And the answer is yes. So we plan to launch an EAP in parallel to the phase 3 study for patients who are not eligible for our phase 3 study. Because as said, we are going to limit the disease duration quite drastically, right? And we do not want to exclude patients who are not eligible to our phase 3 study. However, from a safety point of view, we might have some, of course, exclusion criteria, and these are mainly the exclusion criteria or the warnings and contraindications for celecoxib and ciprofloxacin.
Thank you.
The next question is PrimeC from a compound pharmacy as effective as the product in the trial?
Most probably not, because we used a specific formulation for PrimeC, which actually allows to synchronize the PK curves of ciprofloxacin and celecoxib. So they are fully synchronized in terms of Cmax, Tmax, and so on. So, and this is, I think, very important because we want to, let's say, leverage the synergistic effect Shiran was talking about, right? So by using this specific formulation, I think we are able to increase the synergistic effect of both compounds. And as you have seen, you know, each of the compounds in monotherapy, at least in preclinical trials, using the zebrafish model and human cells on iPSCs, right? We see that the combination is more effective than each of the compounds in mono.
Perfect. Thank you. And the next question was, I know you actually presented, you know, different elements that showed improvements. Were there specifically motor function improvements in the result?
I'm
Yeah,
not
what kind of
sure
results
whether
you-
I understand the question. What do you mean
So
by motor?
just, you know, walking, limb, you know, gait function or fine motors.
Okay. So we have seen actually more or less the same benefit under PrimeC in fine and gross motor function, right? However, the most pronounced effect was on the bulbar function, where we could see a statistical significant difference, right? And we actually gained almost 2 points difference in the bulbar function. But we have seen also quite similar effect on the gross and fine motor function.
Perfect. Thank you.
Okay. The next question is, what is the advantage of PrimeC over just getting separate prescriptions of celecoxib and ciprofloxacin and taking them together? Any side effects, and have you seen elevated liver enzymes or blood pressure?
Yeah, very good question. So as said, I mean, since PrimeC is, let's say, is administered in a specific formulation which synchronizes the PK curves, right? I think it's fair to say that this formulation is more effective than, you know, just compounding the two of them. And also we are using a specific ratio between celecoxib and ciprofloxacin, right? Which is commercially not available. And this, let's say, this dose finding is based on extensive calculation and modeling from our preclinical trials. And the second question was in elevated liver enzymes. No, we haven't seen any, let's say, signals in terms of any hepatic side effects, which is really good. By the way, same for renal function. And as you know, celecoxib has some kind of a history in terms of cardiac adverse events. And for that, we applied a very extensive ECG monitoring. And I'm very happy to say that we haven't seen any, let's say, signals also in terms of cardiac events. And what was the third question, Sylvia? Sorry.
No worries. The
Hypertension.
side effects.
Yeah, hypertension. And no, we haven't seen any, let's say, safety signals in terms of hypertension.
Okay, awesome.
Thank
So
you.
just talking about side effects, I mean, this is so important for a phase 2 study. So what we, the main, let's say, side effects were GI effects, as expected, I would say, right? I mean, this is the beauty when you're working with two so well established compounds, you actually know what you get in terms of side effects. So most of them were GI effects, mild to moderate, right? And all of them were transient.
That's great to hear. So the next question were very much related. Were you trying to prevent ferroptosis, since both drugs come from set of side effects separately, and were patients able to tolerate them, which partially you answered?
Shiran, do you want to talk about ferroptosis?
Yeah, so I think that I mean that's this kind of like a work in progress, and within the paradigm study, I think that's the first time that we dived clinically into trying to assess the specific effect of PrimeC on the iron metabolism. So this is definitely something that we are looking into.
Yeah. And I mean, we know that, an iron deposition, right, leads to neuronal death, right? So, I mean, there are at least one, there are multiple studies, but at least one really good study, an MRI study showing an iron deposition in the cortex of ALS patients, right? So actually you can measure it by I think it was a 7 Tesla MRI machine, right? So we know that, right? And we know also that iron deposition and oxidative stress is interlinked, right? So since ciprofloxacin is an iron chelator, right? It's very, I would say it's very clear that ciprofloxacin has an effect on the iron deposition, but ciprofloxacin with an anti-inflammatory effect might also play an important role in this, in both, let's say, pathological pathways.
Perfect.
All right. The next question is, in your trial and data over the 18 months, were you able to distinguish between fast, moderate, and slow progressors?
Oh, very good question. So we have looked into the 6 months data in fast and slow progressing patients. And what we have seen is that fast progressing patients, right, with an NCAL-3 score of more than 4.5 benefited more than slow progressing patients, right? And Shiran can talk to that. We also looked into NFL and saw also quite an interesting effect, let's say, in correlation with the disease duration. Shiran.
Yeah, so what we've done, and I saw that there was also a question in the chat about NFL. So indeed, we measured the NFL in 6 months, and we do plan now that we concluded the study to measure that again, to have all the time points together. But sorry, indeed, in 6 months, we saw a trend towards a reduction. But as said, NFL show a lot of fluctuation, and the heterogeneity of the disease is quite a challenge when we are talking about statistics. But this is definitely something that we will also measure in 18 months. Nevertheless, as Ferenc mentioned, we tried to analyze the data based on the risk factor and the subpopulation in terms of if it was fast progressors from 13 months from diagnosis, 24, 18, and 12. And as Fran mentioned, when we looked on the shorter time, there we saw a more robust effect in terms of the NFLs.
Great. So the next question is moving on to how is this going to work? Is this something a person with ALS will have to take for rest of their lives?
Yes. So this is actually, I mean, there is no reason to stop on the treatment, right? Because I mean, what you have seen, at least according to the paradigm study, even though the slope of the patients who were constantly treated with PrimeC, it was significantly less steep after 12 months, but there's still a progression, right? So there, from a, let's say, as a neurologist, right, I would say there's no reason to stop the treatment at a certain time point.
Okay. The next question is, when do you believe we could have PrimeC available? What does the timeline look like?
That's a very good question. So we plan, best, I mean, we are biotech, we are always planned with the best case scenario. So 2028.
So the next question, follows that is, until then, could we just take Cipro and Celebrex from Hopewell Pharmacy, which is a specialty pharmacy in the United States, until it's approved?
Yeah. So as a neurologist and physician, I would not recommend out-of-label use. Nevertheless, I understand that patients might be interested in getting both compounds, at a specialty pharmacy, and therefore I would like to ask you to, let's say, discuss this with your treating physician.
Thank you.
By the way, we will never stop and hinder out-of-label use, right? This is, of course, clear. But we are very convinced that our combination is much more effective and safer than let's say, the free combination.
Another question is, did you analyze neurofilament into consideration?
Yeah, so as I said, we did measure the NFL at 6 months, and I share with you that indeed we saw a trend, and when we looked on the sub-analysis that went by time from that diagnosis, we indeed saw that patients with the shorter disease duration gain more effect from PrimeC, and we do plan to measure that now in the 18 months time points, going through all of the tested time points of the study, along with the rest of the biomarkers as Franz showed in our plans.
So the next question is, if if NeuroSense, drug is approved, how do you make sure that it's affordable for people with ALS? Because one of the issues we all had seen with Amylyx was that the price was so high and it was not accessible by insurance.
That's a very good question, but I have to admit, a bit early for us, right? Especially for me as the Chief Medical Officer and Shiran as the VP on R&D, right? So we are, to be honest, we haven't, let's say, discussed this internally that much, right? Of course, we have an idea about pricing, but we haven't done any pricing study. We haven't, let's say, discussed potential pricing with, we haven't discussed this with insurers. This will follow later in, let's say, later in our development cycle, right? Currently, we are
too busy pushing the study forward.
Yeah, we are laser focused on developing the drug and not that much concerned about, let's say, financial things. However, I absolutely understand the concern, right, that the drug might be priced too high, which actually, let's say, might, let's say, causing certain hurdles to get access to the drug. We are very much aware of this, but look, we have not one single commercial person in our company, so we have to grow, and we certainly will tackle that, and hopefully find a good solution for the company, for patients, and the society, the insurers.
Perfect. Thank you.
The next question is, how do people join the study in the proposed phase 3 trial?
That's a very good question. So we haven't, or we are actually currently in the phase to start to identify sites in the US, Canada, Europe, and Israel. By the way, it's going to be a 50-50 split, right? So North America versus rest of world, right? So we're going to have a substantial number of US sites, and we are just currently in the phase to start to identify potential study sites in the US. And as soon as these study sites have been identified, I mean, we are more than happy to share this list of sites with you, with Everything ALS. We will publish it, the sites on clinicaltrials.gov and other, let's say, channels to communicate the study sites.
A sub-question was that how many sites are you targeting in the US? I know you mentioned that you're going in the phase of that, but do you have an estimate regarding the sites?
Well, that's a good question. I would say around 10 sites in North America.
10 sites. And then this is about the inclusion criteria of 18-month disease duration. It seems like it's very limiting for interested participants. What was the rationale to decide the 18-month criteria?
Yeah, as Shiran said, we saw quite a strong signal in neurofilament, right? That PrimeC actually shows a even higher effect in patients with a shorter disease duration. And as I also, there was a question about fast progressing patients. We also saw that patients clinically benefited more. The fast progressing patients benefited more than slow progressors. That's the reason. And to be honest, also to reduce variability in the data.
Great. So if someone's going to be enrolled in phase 3, can they take riluzole and radicava when enrolled in phase 3?
Yes.
Perfect.
The next question is, if a participant was in the Healy trial, would this still be available?
The phase 3 trial?
Mm-hmm.
Yes. So in our current study protocol, we, let's say, of course, we allow patients who have been in another trial, but the patient has to, there must be a washout period of at least 3 months between 2 trials, right? What we cannot allow are patients who are taking any investigational drug in parallel to a study. But this is, let's say, common sense, I would say. So I don't know any study which actually allows that a study patient also takes an investigational drug in parallel to another investigational drug.
Thank you. You mentioned some of the sites, but how can a patient, for example, from Australia be enrolled? Or is there an opportunity for patients in Australia to get the...
So currently we do not plan to run the study in Australia. it's not planned yet, but this might change, right? To be very honest. Yeah. So this is not, let's say, carved in stone. And of course, we are also considering to do an EAP in later, perhaps in 2025 or 2026 in Australia.
Thank you. And I think with that, we are, we answered all the questions, and thank you so much for coming. And, one of the things that you're our consortium members, and you've provided so much to the people with ALS and their community, our community and the families. So bottom of my heart, so thank you. I know that you guys work relentlessly, and any news you have, you want to share with the community, and we've hosted you many times, and each time, you up the bar for everybody else. So thank you for making the time. And as Sarita said, you've been a middle of the night for you, and we very much appreciate your, and we are big cheerleaders, and we are rooting for, the best results here.
Thank you. Thank you so much.
Keep doing the amazing things that you do.
Thank you so much. We appreciate you, and we are on your team. So use us when you can.
We will do.
And once again, thank you for your time.
Thank you.
We know how precious it is. Please know that we're here for you. Whatever portion of Everything ALS you would like to use, it's there with a smile. If you ever need somebody personally to share your concerns with, look up the Pathfinders. And with that, let's all say goodnight to each other. Good night.
Bye, guys.
Thank you, Gazella. Bye for now.
Okay.